Good evening friends. Today we shall discuss about capillary electrophoresis. Before that, I request all of my friends, please subscribe my YouTube channel and also watch, like and share my videos with your friends. Capillary electrophoresis is a powerful technique that employs narrow-bore capillary tubes for achieving high-efficiency separations of both small and large molecules. These separations are facilitated by the use of high voltage. There are different modes of capillary electrophoresis. Number 1. Capillary zone electrophoresis, also known as free solution capillary electrophoresis. By this technique, biomolecules are separated based on the differences in their size and net charge. It is the most widely used method of capillary electrophoresis. Second one is micellar electrokinetic capillary chromatography. It is a technique that separates neutral compounds by using surfactant micellars. Number 3. Capillary isoelectric focusing, by which biomolecules are separated according to their isoelectric points. Fourth one is capillary gel electrophoresis. In this technique, a suitable gel is used as the stabilizing medium. The porous gel serves as a molecular sieve that separates sample components based on their size. Fifth one is capillary electrochromatography that involves both electrophoretic and chromatographic methods. This figure illustrates the basic components of a typical capillary electrophoresis system. Number 1. A narrow-bore capillary tube with an internal diameter of about 10 to 100 micrometer. Its external diameter is about 400 micrometer. The length of the capillary tube is between 10 and 100 centimeter. The most popular capillary material used is fused silica that is transparent to the UV and visible light. The tube can be externally coated with a polyamide material to increase the flexibility. Finally, the capillary tube is enclosed in a thermostatically controlled environment for temperature control. The second component are two buffer reservoirs filled with a suitable buffer solution. The buffer is also known as carrier electrolyte or background electrolyte. The purpose of using a buffer is to maintain the pH and conductivity during electrophoretic separation. A controlled pH is also crucial for maintaining a constant net charge on the biomolecules, thereby maintaining the electrophoretic mobility. Another component is a high voltage power supply. The fourth components are two electrode assemblies. The fifth component is a sample introduction system by which very small amount of sample is injected into the buffer-filled capillary tube. There are two different methods of sample application. One is pressure injection and the other is high voltage injection. One of the most important components of capillary electrophoresis is the detector. Detection schemes for capillary electrophoresis includes measurement of UV absorption, refractive index and fluorescence. Electrochemical signals and conductivity as well as radioactivity from radioisotopes have also been measured. Measurement of UV absorption at a chosen wavelength is the most widely used method of sample detection. Finally, the detectors send a signal to a output device which can be an integrator, a recorder or a computer. This figure illustrates the process of capillary electrophoresis. After very small amount of sample is injected into the buffer filled capillary tube, high voltage up to 50 kilovolt is applied across the tube. Under the influence of the electric field, charged molecules will migrate towards the oppositely charged electrodes. Positively charged molecules will migrate towards the cathode, whereas negatively charged ones towards the anode. One of the fundamental processes that drive capillary electrophoresis is electroosmosis. Few silica capillaries contain silanol groups that become ionized in buffer. At a pH greater than 2 or 3, the silanol groups become ionized to form negatively charged silanate ions. Cations present in the buffer solution are attracted towards the silanate ions. Some of these cations are tightly bound to the silanate ions. This forms a fixed layer. As the negative charge on the capillary wall is partially neutralized by the buffer cations, the solution adjacent to the fixed layer contains more cations than anions. This forms another layer called diffuse layer. Fixed layer and diffuse layer together form the double layer. Under the influence of the electric field, these cations will migrate towards the cathode. As these cations are solvated, the bulk solution is also pulled along towards the cathode. This movement of the bulk solution through a charged surface towards the cathode under the influence of an electric field is called electroosmosis. Although sample components are separated based on their electrophoretic mobility, it is because of electroosmosis, all of them including both positively and negatively charged molecules and also neutral species, all of them will migrate towards the cathode. The rate of electroendosmotic flow is much greater than the electrophoretic velocity of analytes. It is because of the higher electroosmotic flow, all ions, regardless of their charge sign and neutral species, are carried towards the cathode. Positively charged molecules will reach the cathode first. This is because the combination of electrophoretic migration and the electroosmotic flow cause them to move faster. Positively charged molecules will be followed by neutral species, which in turn will be followed by negatively charged molecules. As the separated molecules approach the cathode, they pass through a viewing window where they are detected by an ultraviolet monitor. The monitor then sends a signal to an integrator, a recorder or a computer. The signal thus generated is plotted against migration time to form an electrophotogram. Typical run times are between 10 and 30 minutes. In this way, sample components are separated by capillary electrophoresis. One of the major advantages of capillary electrophoresis is that it consumes only microliters of the reagent and only nanoliters subsample are needed for analysis. This technique can be used to separate a wide spectrum of biomolecules including amino acids, peptides, proteins, DNA fragments and nucleic acids, as well as any number of small organic molecules including drugs and even metal ions. Other applications are identification of point mutations in DNA and separation of chiral compounds. Friends, this is all about capillary electrophoresis. For more updates, subscribe my YouTube channel, Sam Biology Class.